We've spent some time discussing Mendel and his ideas and how the basics of genetics work. I want to talk about the actual physical structure of DNA, how it's replicated in S phase of interphase, and later on how it actually serves as the instructions for the cell. Because you always hear, oh, DNA is like the template, it's the instructions that tell the cell how to do everything. And we're going to discuss how that actually works. So again, this is us getting into chapter 9. So this is Watson and Crick, who were two of the gentlemen who discovered the structure of DNA. I think perhaps fittingly, Rosalind Franklin didn't make it into this photo. Rosalind Franklin was the individual that did the X-ray crystallography for Watson and Crick, which actually resulted in a picture that looks something like this that allowed them to figure out, oh, hey, this is a double helix. Here's what's going on. She was not awarded the Nobel Prize, but these two were. And that's just kind of a product of the time. She died of radiation sickness as a result of her work as an X-ray technician, or X-ray crystallographer, I should say. And to this day, she largely doesn't receive much of the credit that she deserves. You have the American and the Englishman, Watson and Crick here, who worked with Rosalind Franklin. I'm a big fan of Rosalind Franklin, by the way, so that's a name you should know. So we've already discussed kind of at length the structure of nucleic acids, and so you should probably already know what's going on with DNA. Again, I'm not going to ask you for like specifics of chemical structures, like where are the carbons located, but you need to know that DNA is composed of a sugar, which is attached to a phosphate, which will then be attached to the next sugar. And it's also, the sugar is also attached to the nitrogenous base that makes up the letter code that is your DNA. So again, the double helix is formed of repeating patterns of sugar and phosphate, and the nucleotide bases here, this being a G and this being a C, are connected through hydrogen bonds. Where have we seen hydrogen bonds before? With water. Hydrogen bonds are a weak bond that we're actually, and I say we, I mean all of your cells, are actually going to be able to unzip using an enzyme. It's actually going to be able to break these hydrogen bonds so that it can either replicate or so it can read from one of these individual DNA strands. So we've talked a little bit about DNA and we've talked a little bit about RNA, but they are two different molecules. The reason that they're different, and again, I'm not going to ask you for chemical specifics, but ribose is ribose has an oxygen, and deoxy, as the name would imply, is missing that oxygen on this 2' carbon. Okay, so chemically, they're very similar to one another, but there is at least one subtle difference. We're going to discuss this later on, but in DNA, you have A's, G's, T's, and C's, and in RNA, you have A's, G's, C's, and U's. They substitute thymine with uracil, but we'll get to that. We'll cross that bridge when we come to it. But you should know there is a chemical difference between DNA and RNA. A subtle one, but it is there. And again, this is something we've already discussed maybe a couple of times, but in eukaryotes, they have a region where their DNA all hangs out, called the nucleus, that has a physical membrane around it. In prokaryotes, there's just kind of an area where the DNA is more likely to be. There's no membrane around it. It's just in the middle of the cell. We call that the nucleoid. So again, as I was discussing in the meiosis and mitosis section, if you're zoomed all the way in on a piece of DNA, it's going to look like this double helix structure. And most of the time your DNA is spent sort of unspooled, like you just took a bag of yarn and gave it to the cat or threw it everywhere in a room. But it is sort of organized by wrapping up around these things called histone proteins. Okay, it's wrapped up around these things called histone proteins. And those histone proteins are going to further wrap around one another and form sort of like another layer of organization. And when your chromosomes are actually condensing, they will form something that looks like this. And you can tell this piece of DNA has already gone through S phase. And you can tell that because there are two sister chromatids here. Okay, the point that I'm trying to illustrate here is that, again, most of the time your DNA is unspooled throughout the cell, and during certain times it's actually going to condense. But the reason that it's unspooled and that it needs to be unspooled is because when we try to replicate it or when we try to read from it, if it's all tightly wound like this, that's going to be impossible. So the next thing about DNA is that there are base pairs that are complementary to one another. So whenever you see an A on one side of the ladder of the double helix, you're going to see a T on the other. And this is something you should know. You should know that A is supposed to always be matched with T in DNA, and that G is supposed to always be matched with C in DNA. Okay, these are complementary base pairs. And we're going to talk about DNA replication, but it is referred to as semi-conservative because of the way that it actually winds up working. So here's one strand of DNA, so that would be like this one here. Here's another strand of DNA, that would be like this one here. And what's going to wind up happening is that is going to get opened up. The hydrogen bonds between those complementary base pairs are opened up, and enzymes are going to go through and replicate it. But you'll notice here, we start out with two complementary strands or one segment of DNA, and we replicate it. And so you wind up with two semi-new segments of DNA. And when I say semi-new, you can see that this gray one started out on our original strand. And this blue one was synthesized by those enzymes. But here are our two new pieces of DNA that still contain the old original. Okay, for this reason, DNA replication is referred to as semi-conservative, because you're winding up with some of the old strand, with some of this old segment of DNA in every new piece that you create. DNA replication is referred to as semi-conservative. We're going to talk about the specifics of replication in the next lecture.